Germline Characterization of Genes Associated with Spermatogenesis and Embryonic Developmental Competence in Azoospermic Men. Infertility affects about 15% of humans of reproductive age. Of this population, about 33% of the cases are attributed to pure male factor infertility. The first evaluation used to screen men undergoing fertility treatment is a semen analysis. This straightforward test visits a variety of aspects such as abstinence period, concentration, motility, morphology, and the quantification of foreign cells. In a normal semen analysis, spermatozoa are immediately identified upon initial observation, as seen here. However, when the semen analysis reveals no spermatozoa, an extensive sperm search is performed. When no spermatozoa are identified, the patient is diagnosed as azoospermic. Azoospermia affects 1% of all men and 15% of men with infertility. 40% of these cases can be attributed to obstructive azoospermia, which could be due to a congenital defect of the vas deferens or of the testis. It can also be acquired by vasectomy, injury, or infection of the reproductive tract. The remaining 60% can be attributed to non-obstructive azoospermia, of which the etiology is not always clearly known. There are three main types of non-obstructive azoospermia. The first is hypospermatogenesis, in which there is low sperm production and scant numbers of sperm are identified in the seminiferous tubule. Luckily, this is the most common type of non-obstructive azoospermia, but there are other situations, such as maturation arrest, where tubules have immature germ cells that do not progress to spermatozoa, and Sertoli cell-only syndrome, in which there aren't even germ cells present in the seminiferous tubules from which to generate sperm. Therefore, sperm retrieval by surgical intervention is necessary. In these men, it is important to understand the etiology of this condition by carrying out genetic analyses. One study, performed on the peripheral blood of infertile men, confirmed that male factor is associated with chromosomal abnormality. Nakamura and colleagues found that azoospermic men had an extremely high incidence of autosomal as well as gonosomal aneuploidy. They concluded that cytogenetic abnormalities can affect sperm parameters, as azoospermia was shown to create a major problem with the peripheral karyotype. Other studies on the peripheral blood observed wide deletions in patients with lower sperm concentrations. However, in spite of this, these patients still had a very satisfactory clinical pregnancy rate. While these studies are informative, we were interested in assessing the actual spermatozoa of these azoospermic men. Genetic assessments on spermatozoa was commonly carried out by cytogenetic assays such as fluorescence in situ hybridization, or FISH. And although the assessment by FISH has proven useful, it is important to recognize that we are still limited by the number of chromosomes that we can assess for, as well as the possibility for hybridization failure and FISH error. Lastly, we are limited by the concentration of spermatozoa, particularly in these surgical specimens. Therefore, to properly assess the genetic profile of the male gamete, a more sensitive approach is needed. Next-generation sequencing is a more thorough and accurate technique for genetic evaluations. Once extracted and amplified, spermatozoal DNA can be assessed for not only aneuploidy, but the presence of gene mutations. Here we can see that when we compare the sperm aneuploidy assessed by FISH, depicted in various colors, to the assessment by NGS, shown here in green, there is a dramatic difference, demonstrating the sensitivity of NGS. The concern regarding the use of surgically retrieved spermatozoas has actually been around for a while. Early studies utilizing FISH on spermatozoa from ejaculated and surgical sources concluded that surgically retrieved spermatozoa possess higher aneuploidy than their ejaculated counterparts. We first sought to disprove these previous findings, since the incidence of miscarriages and abnormalities in children conceived from surgically retrieved spermatozoa was significantly lower than those conceived from ejaculated specimens, and also lower than the national threshold of the U.S. We revisited the aneuploidy profile of surgically retrieved sperm by comparing it with their ejaculated counterparts. We were surprised to find that the FISH aneuploidy was indeed not higher in the epididymal and testicular sperm than in the ejaculated sperm, not for the gonosomal and not for the autosomal. When we performed NGS, we confirmed that the surgically retrieved sperm aneuploidy was certainly not greater than that of ejaculated sperm, as it was evidenced in past studies. In fact, with this particular assessment, it was significantly lower. And we were curious to repeat the analysis within the same individuals, so we assessed patients who had high DNA fragmentation in their ejaculates, who would therefore agree to undergo a testicular biopsy. Our assessment of ejaculated and testicular sperm aneuploidy from within the same patients found that although FISH was not able to evidence a significant difference, the NGS was able to confirm that testicular sperm aneuploidy is certainly not higher than that of the ejaculated specimen. Moreover, while there were no pregnancies resulting from these patients' cycles using ejaculated spermatozoa, cycles using their testicular spermatozoa resulted in successful clinical pregnancies. So, while this assessment was done on a limited number of observations, the findings evidence that surgically retrieved specimens are safe to use. In the current study, our aim is to sequence spermatozoal DNA from Azoospermic men to identify germline mutations related to the etiology of their infertility and ability to support a pregnancy. Men undergoing infertility treatment with their partners were screened by semen analysis according to WHO criteria. In the event that no spermatozoa were identified after a repeat semen analysis and extensive search of the sample, the patient was referred to a urologist for surgical sperm extraction. Those men diagnosed with obstructive azoospermia, acquired by vasectomy, underwent epididymal sperm aspiration, while those with non-obstructive azoospermia underwent testicular biopsy. The surgically retrieved spermatozoa were used for ICSI, and a small portion of the specimens were assessed by DNA sequencing. Following the couple's cycle outcomes, a genomic analysis was carried out in which we evaluated the germline mutations and gene imbalances according to the patient's pregnancy outcomes. Of the 27 couples included in this study, 17 OA men underwent surgical sperm retrieval, while 10 NOA men underwent testicular biopsy. These procedures were all successful in retrieving spermatozoa for ICSI. The obstructive cohort underwent 17 ICSI cycles, resulting in a 47% clinical pregnancy and delivery rate, while the non-obstructive cohort underwent 10 ICSI cycles, yielding a clinical pregnancy and delivery rate of 70%. First, we assessed the overall aneuploidy of the spermatozoa for each cohort by NGS. We did not observe a significant difference in sperm aneuploidy between the obstructive and non-obstructive groups. We then assessed the genes that were present in all of the patients from the OA and NOA cohorts, respectively. In the OA group overall, only 3 common gene mutations were identified, shown here in green. These were mainly housekeeping genes that were not related to spermatogenesis or embryo development. In the NOA cohort, however, 5 gene mutations were found that were involved in RNA transcription, apoptosis, and basic spermiogenic functions. Now, to graphically represent the duplications and deletions on these genes to compare the two categories, we see that the genes found in the NOA cohort display higher duplications and deletions than those from the OA cohort. In addition, we notice that AP5M1, related to apoptosis, was characterized by the most duplications, followed by APOE, a gene involved in basic spermiogenic function. We then grouped the obstructive patients according to their ICSI pregnancy outcomes. For those able to reproduce, all shared a mutation in a gene mainly responsible for transcriptional regulation during spermatogenesis. Here is a graphic representation of the duplications and deletions found on this gene. The results of a differential expression analysis by RNA sequencing also confirmed an imbalance of this gene. And as for those patients unable to generate a pregnancy, a common mutation was found on a gene involved in DNA replication. When a genomic assessment was carried out, we saw that not only were more deletions found on this gene, but it was significantly underexpressed as well. The non-obstructive patients were then grouped according to their ICSI pregnancy outcomes. While the fertile cohort displayed a single mutated gene that was related to stem cell lineage differentiation, those unable to reproduce had 6 gene mutations involved in spermatogenesis, 4 related to apoptosis, 2 for acrosomal function, and 8 for early embryonic development. We also performed a pilot gene expression analysis on NOA patients unable to reproduce. This differential gene expression analysis by RNA sequencing revealed 8 significantly underexpressed genes in the NOA infertile cohort. Their functions included, but were not limited to, DNA repair, export of mRNA, and apoptosis. In conclusion, surgical sampling in patients with azoospermia is successful in granting a pregnancy. Contrary to earlier studies, aneuploidy in surgically retrieved spermatozoa, even by NGS, is negligible. Still, not all azoospermic men are able to achieve a pregnancy with their partners. According to our findings, the reason seems to be that these men possessed key reproductive gene mutations, especially those involved in embryo development. Furthermore, differential gene expression analyses identified imbalances, particularly in genes related to DNA repair. Therefore, genomic profiling of infertile men can provide valuable information on the etiology of their azoospermia as well as their reproductive potential. Thank you.